Hello everyone, welcome back to the class. So in our previous class we discussed about cell cycle checkpoints. So these cell cycle checkpoints functions to ensure that incomplete or damaged chromosomes are not replicated and passed on to daughter cells. So there are many cell cycle checkpoints, but there are major three, that is cell cycle checkpoints, DNA damage checkpoints, spindle assembly checkpoints. So this cell cycle checkpoint was hypothesized by Hartwell and Wienert. They said the existence of checkpoint pathways by which an uncompleted cell cycle event sends an inhibitory signal to later events. So it decides whether to pause, proceed or exit the cell cycle. So cell cycle checkpoint, which is the first one, ensures the fidelity of the cell division. These checkpoints verify whether the process at each phase of the cell cycle have been accurately completed before progression into the next phase. So an important checkpoint in G1 phase we discussed yesterday was the start in yeast and restriction point in mammalian cells. This is the point at which the cell becomes committed to DNA replication and completes a cell cycle. So it is during the G1 phase that the cell integrates mitogenic or growth inhibitory signals and makes the decision to proceed, pause or exit the cell cycle. And we also discussed about the second checkpoint, that is spindle assembly checkpoint, which is also known as mitotic checkpoint. It monitors the correct attachment of chromosomes to the mitotic spindle. It prevents segregation of sister chromatids until they are properly aligned on the metaphase plate. Chromosome segregation is monitored to prevent aneuploidy. Microtubule from opposite spindle poles attached to the kinetochore of a metaphasic chromosome. So this process is monitored by the spindle assembly checkpoint, and the tension develops at the cross-paired kinetochore. So it by determining the mitotic force or the tension which is developing in the paired kinetochore, the kinetochore will harbor a different type of error correction mechanism, and these error correction mechanism is mediated by Aurora B kinase, that is serine 309 kinase, which eliminates incorrect microtubule attachment by sensing whether or not the centromere is under tension. So we discussed about the three different types of attachment of kinetochore, which kinetochore distinguishes and harbors the error correction mechanism. One is amphitelic, where sister kinetochores are anchored to microtubules from opposite poles, which is correct attachment. Erroneous monotelic, where only one kinetochore is anchored to the microtubule, and syntelic, where both kinetochores are anchored to microtubules emanating from the same pole, which is also a wrong or erroneous attachment. Mechanisms that monitors and responds to kinetochore microtubule attachment is SAC, which is a complex, spindle assembly checkpoint complex. It is composed of many proteins that are conserved from yeast to mammals. So the core components include MAD1, MAD2, MAD3. In mammals it is BUB1 and BUB3. MAD stands for mitotic arrest deficient. BUB stands for budding uninhibited by benzimidazole. So during metaphase, MAD1 and BUB1 monitors the proper spindle microtubule kinetochore attachment or tension. So until the proper attachment is not formed, APC-C's activity is inhibited by SAC. So when there is a problem, SAC is activated. What happens when SAC is activated? MAD2 interacts with MAD1 at the kinetochore. There occurs a conformational change in MAD2, which promotes formation of mitotic checkpoint complex, that is MCC. This MCC is a complex made up of MAD2, MAD3, BUB3 and APC-C coactivator protein, that is CDC20. All together forms MCC. So if there is a problem in SAC, MAD2 prevents CDC20 from binding and activating APC-C, resulting the inactivation of APC-C until the problem is not solved. Once kinetochore has established the stable correct amphitallic attachment to the spindle, SAC is silenced and APC bar C is activated at last. However, there was another attachment, that is merotellic attachment, where a single kinetochore is attached to the microtubule from both poles, that can go uncorrected as they do not activate the SAC, which leads to the segregation errors. So SAC failure results in aneuploidy in the daughter cells. And the inhibition of CDC20 is elevated, and APC bar C anaphase promoting complex is activated. SAC failure causes aneuploidy in daughter cells. So in this image you can see the spindle checkpoint process, or the mechanism taken by the spindle checkpoint. Signaling by unattached kinetochore. Signaling by unattached kinetochore stops the cell from entering anaphase until all chromosomes have made a proper bipolar spindle attachment. This indicates SAC is activated. As long as there is a chromosome that is not properly attached to the spindle, the cell does not enter the anaphase stage. The cell enters the anaphase stage about 20 minutes after chromosome attachment is complete. In the cell with a persistently mal-oriented chromosome, anaphase entry is delayed. If the unattached kinetochore is destroyed with the unattached kinetochore or the free kinetochore, if it is destroyed with the high powered laser, the cell enters anaphase about 20 minutes later. This proves that the unattached kinetochore sends an inhibitory signals. The unattached kinetochore sends a signal via MAD2. Okay, MAD2 protein that ultimately inhibits the APC bar C, blocking the degradation of cyclin B and securin, and thereby blocking the transition into the anaphase. So MAD2 protein shown in its complex with MAD1 here. So this was about spindle checkpoint. And also we discussed about the DNA damage checkpoint, which involves the signal transduction pathway. This signal transduction pathway is induced by a DNA damage. This blocks cell cycle progression until the DNA is properly repaired. There are 3 DNA damage checkpoints based on the stage in which they occur. That is G1 bar S checkpoint, which inhibits the G1S transition in cells that have not yet been activated. Intra S phase checkpoint, it prevents the initiation of DNA replication at origins that have not yet been activated. And next is G2 bar M phase checkpoint that inhibits the entry into the mitosis. So there is another checkpoint which is a replication checkpoint. It is a specialized branch of DNA damage checkpoint. It monitors the fork problems and triggers cellular response aimed to the preservation of genomic integrity. So in today's class we will be learning about DNA damage response signaling pathway. We know that DNA damage checkpoint will block the cell cycle progression until the DNA is properly repaired. The cell undergoes different types of signaling pathway which with the response of DNA damage. So the cell will not leave this DNA damage response. By sensing the DNA damage, it undergoes different types of signaling pathway, that is signal transduction pathway, in order to induce this repair mechanism, in order to start the repair mechanism in which DNA can be repaired and the cell cycle can be resumed. So let's know about the 2 different pathways, that is ATM pathway and ATR pathway. The first pathway of signaling pathway, that is ATM pathway. ATM pathway. What is this ATM? ATM stands for Ataxia, Telangiectasia, Telangiectasia mutated. So ATM stands for Ataxia Telangiectasia mutated. So this is a closely related kinase. So it is a closely related kinase that is activated by DNA damage. So this kinase, to be particular, it is a serine threonine protein kinase. It will also phosphorylate serine threonine residues. So ATM pathway, ATM stands for Ataxia Telangiectasia mutated, and it is a closely related kinase that is activated by DNA damage. So it is a serine threonine protein kinase, and it is activated by DNA damage. What is its function? So both ATM and the other one, that is ATR, are activated by DNA damage. The DNA damage may be of two types, exogenous or endogenous. DNA damage may be two types, exogenous and endogenous. Exogenous means the DNA damage is caused by external factors, that may be by ionizing radiation or by chemical mutagens. With the help of or with the effect of this ionizing radiation, chemical mutagens, which are the external factors which cause DNA damage. So that damage may be exogenous. Endogenous is where the DNA damage is by internal factors, that is by metabolic products or stalled replication fork. If there is any defects in fork, that may cause DNA damage. So DNA damage may be of two types, that is exogenous, which is causing by the external factors, and endogenous, which is causing by the internal factors. In response to DNA damage, eukaryotic cells activate elaborate cellular networks. So if there is a DNA damage, irrespective of the factors which has caused this DNA damage, this will initiate a elaborate cellular response. That responses are collectively termed as DDR. DDR stands for DNA damage response. In response to the DNA damage, eukaryotic cells activate different types of cellular networks, which collectively termed as DNA damage response. So these DNA damage response, along with the pathway which we discussed, ATM pathway or ATR pathway, that will lead to, all this will lead to, first DNA repair. It tries to repair the errors. Then it arrests the cell cycle. So cell cycle arrest. Then if nothing works, so then it will lead to apoptosis. Apoptosis is nothing but a programmed cell death. If there is no chance of repairing anymore, if there is no chance of recovering the lost DNA by the damage, so then the cell will initiate the apoptosis process. So let us know about ATM pathway now. ATM pathway. We know that ATM pathway is a what? Is a Ataxia, Telangiectasia mutated pathway, which is a DNA damage response signaling pathway, and ATM is a closely related kinase that are activated by the DNA damage, and these are serine threonine protein kinase and are activated by the DNA damage. So this ATM pathway or ATM responds particularly to DNA double-stranded breaks. DNA double-stranded breaks. So it will particularly respond to the DNA double-stranded breaks. At the molecular level, so DDR pathway, that is DNA damage response pathway, contains several key components, that is DNA damage sensor, which senses the DNA damage, signal transducers, which initiate the signal transduction pathway, and effectors. So various types of DNA damage can be detected. Let me write down the major components of the DDR first. Components of DDR. Irrespective to the pathway which is the cell undergoing, that is ATM or ATR, so there are several response, that is DNA damage response. This DDR pathway contains several components, that is DNA damage sensors. With respect to ATM pathway, the DNA damage sensor detects the double-stranded break. Then it initiates signal transduction. Who is initiating? Signal transducers. Signal transducers. Then the last component is effector. Keeping all this point in mind, let me start the ATM pathway now. ATM pathway. We know ATM pathway responds to DNA double-strand break. Consider there is a DNA. I'll start again. So ATM pathway there is a double-stranded DNA where there is a break here. Okay, break here. First, the sensor senses this DNA double-stranded break. The sensor of ATM is mRN. Okay, just remember this mRN abbreviation. The mRN complex acts as a double-strand break sensor in the ATM pathway. This mRN is a complex. Remember this also. mRN it's a complex. This recruits ATM at the double-strand break site, and the interaction with mRN complex activates ATM kinase. So the interaction between the ATM and mRN complex will activate the ATM kinase. This activation of ATM kinase phosphorylates the kinase CHK2. Okay, phosphorylates the kinase CHK2, and there is initiates the signaling which is converging on downstream effectors. So which initiates signaling converging on the downstream effector. So now comes effector. The effector in this ATM pathway is P53. P53. So DDR mediated cellular outcome may be cell death by apoptosis or checkpoint arrest. So in ATM pathway, so there is checkpoint arrest. So after the downstream effector, or with the help of the signaling converging on downstream effector, that is P53, so the checkpoint is arrested. So there is checkpoint arrest. So this is how the ATM pathway works. So if that DNA damage can be repaired, so then the ATM will repair it. If not, it will lead to apoptosis, that is a programmed cell death. So this was about ATM pathway. Remember, mRNA is a sensor here. Okay, signal transducer is Chk2, which is initiating the signaling. Then the effector is P53. That effector will halt the checkpoint, will tell that there is a problem here, then we should arrest the cell cycle. So this was about ATM pathway. So next we'll discuss about ATR pathway. ATR pathway. This ATR stands for Ataxia, same, Ataxia, Telangiectasia, same, Telangiectasia, and Rad3-related. So ATR stands for Ataxia, Telangiectasia, and Rad3-related. So this is a serine threonine protein kinase, and it is a member of phosphatidyl inositol 3' kinase like kinase. So this belongs to PIKK family. PIKK family, both ATR and ATM. So PIKK full form is phosphatidyl inositol 3' kinase like kinase. So this kinase belongs to PIKK family. This ATR pathway is also, the ATR is also activated by DNA damage, irrespective of the DNA damage source, that may be exogenous or endogenous. Now we'll know about the ATR pathway. ATM pathway will respond to double-stranded break, we know. ATR pathway will respond to single-stranded breaks. Single-stranded breaks, and it is associated with the wide range, or it responds with wide range of DNA lesions. DNA lesions which is caused by or associated with the DNA replication. We know when there is a DNA damage, eukaryotic cells activate elaborate type of cellular networks which is collectively termed as DNA damage response, DDR. In molecular level, this DDR pathway contains several components that also we discussed, that is DNA damage sensors, signal transducers and effector molecules. With the help of all these components, ATR pathway will repair or will arrest the checkpoint. Let us see now. ATR pathway, what does it do? So it will sense or it will respond to single-stranded break. So I'll write the double-stranded DNA itself and I'll make a single strand break. Okay, single strand break. It will be good enough to draw the break here. Okay, at this there is a single strand break. Here the sensor RPA plays an important role. RPA stands for replication protein A. This replication protein A is a single-stranded DNA binding protein which functions as a sensor in the ATR pathway. RPA binds to the single-stranded DNA generated at the site of stalled replication fork. Okay. With this, the another apical kinase, that is ATR interacting protein. In short, we'll call it as ATRIP, or you can call ATRIP. ATRIP stands for ATR interacting protein. Okay, so ATR interacting protein binds to the RPA single strand DNA complex. When RPA is bound with the single stranded break, it forms a complex. Okay, it forms a complex. RPA and single stranded complex will bind to ATRIP binds to RPA single stranded DNA complex in order to recruit ATR. Now this will recruit ATR to single stranded DNA which activates the downstream kinase. That is CHK1, which is known as effector. Okay, so eventually DDR signaling spreads away from the damaged locus owing to the engagement of the diffusible kinase CHK2, which is phosphorylated by ATM. So this effector CHK1 is phosphorylated by ATR. In the case of ATM pathway, ATM will phosphorylate which effector, that is CHK2. Okay. In ATR pathway, CHK1 is phosphorylated. In ATM pathway, CHK2 is mainly phosphorylated. After this phosphorylation, it signals the converging on downstream effectors. That is, in case of ATM, it was P53, but in this case, the effector molecule is CDC25. So this DDR mediated cellular outcomes may be cell death by apoptosis or transient cell cycle arrest followed by repair of DNA damage and resumption of proliferation, or cellular senescence caused by the persistence of unrepaired DNA damage. So to summarize the two pathways, I'll write it in table format. I'll write ATM pathway here and ATR pathway here. Both ATM and ATR pathway are a closely related kinase, serine 309 kinase, which belong to PIKK family. Both this pathway is activated by DNA damage. So the DNA damage may be caused by exogenous action or endogenous action. Okay, so the eukaryotic cell will activate the elaborate cellular network, which collectively termed as DNA damage response, DDR. These signaling pathways lead to DNA repair, cell cycle arrest, and eventually apoptosis. This ATM responds to double-stranded break. ATR pathway responds to single-stranded break. Okay. At the molecular level, the DNA damage response involves three components: DNA damage sensors, signal transducers, and effectors. So, first the sensor comes into picture. The sensor in ATM pathway is mRNA. mRNA is a sensor which binds to the DNA damage site, that is double-stranded break. Here in ATR, the sensor was RPA. RPA binds to the damaged portion. Then ATM binds to mRNA, will activate CHK by phosphorylating it, just phosphorylating it. Activation of CHK2 is by phosphorylation by ATM. And here there is another downstream there is another molecule, that is ATRP. ATR interacting protein. This ATR interacting protein binds with the RPA and single-stranded DNA complex. Okay, will recruit ATR to the single-stranded DNA, and that activates CHK1 by phosphorylating it. And after that it will signal the converging downstream effectors, such as in ATM, which was the effector P53, in ATR, which was the effector CDC25, for further action, for further outcome, whether for apoptosis, whether for cell cycle arrest, whether for cellular senescence. It depends on the situation, and it depends on the depth or frequency of the DNA damage. So this was about DNA damage response repair pathway. So that is ATR pathway and ATM pathway. In our next class, we will be discussing about role of RB protein in cell cycle regulation. Before ending this class, let us see some images related to this concept. See, DNA damage response signaling pathway, that is ATM, Ataxia, Telangiectasia mutated, ATR, Ataxia, Telangiectasia and Rad3-related, are closely related kinase, that is serine 309 protein kinase. Both these pathways are activated by DNA damage, whether it may be exogenous or endogenous. They belong to PIKK family, that is phosphatidylinositol 3' kinase like kinase. In response to DNA damage, eukaryotic cells activate elaborate cellular networks, collectively termed as DNA damage response, DDR. So they contain several components like sensors, transducers, effectors and response. Here you can see the sensor, ATM dimer, ATM monomer, ATR plus cofactors bind single-stranded DNA. Then transducers molecules are CHK2, P53, CHK1. Then effectors, cell cycle proteins, DNA repair proteins. Then the response may be apoptosis, cell cycle arrest and or DNA repair. ATM pathway. ATM pathway responds to double-stranded break. ATR responds to wide range of DNA lesions, especially those associated with DNA replication. Sensor proteins detects the DNA damage. The sensor protein of ATM pathway is mRN. Okay, mRN. Double strand break sensors in ATM pathways recruit ATM at double strand break sites. And interaction with mRN complex activates ATM kinase, results in phosphorylation of kinase, that is CHK2. See here, double stranded break where ATM is recruited by mRN, okay, and then that phosphorylates CHK2. ATR pathway, the sensor molecule is RPA, replication protein A, which is a single strand DNA binding protein, and it is a sensor in ATR pathway. RPA plus single stranded DNA forms a complex at the site of stalled replication fork. ATR interacting protein, that is A-trip, binds with the RPA single stranded DNA complex which recruit ATR. Okay, ATR is active, activates the downstream kinase CHK1. The DDR signaling spreads away from the damaged locus owing to the engagement of diffusible kinase CHK2 and CHK1, with signaling converging to the downstream effectors such as P53. In this case, it is CDC25, which is phosphorylated. DDR mediated cells outcome is maybe cell death or apoptosis or degradation or rapid cell cycle arrest. So this was about the ATR pathway. See here, ATM and ATR pathway. So this is already dealt. So in our next class, we will be learning about role of RB protein, role of P53 in cell cycle regulation. Thank you.